Hi friends, so now I'm back with my next video, and in this video we are going to talk about pentose phosphate pathway. It is also known as hexose monophosphate pathway because it's the alternative to the glycolysis, like the glucose that is entered the glycolysis. After get converted to hexose 6 phosphate, it enters into another route, which is called the pentose phosphate pathway. So please make sure to like, share and subscribe my channel. So what is a pentose phosphate pathway? It is an alternative pathway to glycolysis and the TCA cycle, which is for the oxidation. So for the glycolysis, glucose enter and produces pyruvate, which enters the TCA cycle. But in the pentose phosphate pathway, instead of producing pyruvate, glucose from glucose 6 phosphate which enters the pentose phosphate pathway to release NADPH and the ribose phosphate, which are used for the synthesis of nucleotides. It is also known as hexose monophosphate shunt or phosphogluconate pathway as well. And it is a shunt of the glycolysis, like instead of going to the glycolysis, the substrate is shunted towards the another pathway, and it occurs mostly in the cytoplasm in both eukaryotes and prokaryotes. And while it involves the oxidation of glucose, which means that glucose is oxidized to produce, but its main role is anabolic and rather than catabolic, because the NADPH which is released is mainly used for the synthesis of lipids, and also the ribose phosphate which is produced is used for the synthesis of nucleotides. So its major role is anabolic rather than catabolic. So hexose monophosphate pathway, the trade-off whether to go like the glucose will enter the glycolysis or the hexose monophosphate pathway is decided by the concentration of NAD plus to NADPH. So if the concentration of NADPH is higher, then there is no need for this pathway. The glucose can enter the glycolysis pathway. But if the levels of NADPH are less, then there is need for NADPH for the synthesis of fatty acids, lipids, and for also for the nucleic acid. So then the glucose enters the this hexose monophosphate pathway. So it is decided by the ratio of NAD plus to NADPH. And as I have talked about the NADPH which is produced, it is used for the synthesis of lipids, ribose for nucleotides, and nucleic acid synthesis as well. And it is mainly useful in the adipose tissue, liver, because this is the place where the fatty acids are synthesized, right? NADPH is needed in all this major tissue for their proper functioning. So there are basically different phases in this hexose monophosphate pathway. So there are two different phases. First is called the oxidative pathway, oxidative phase, because in this phase, the glucose is oxidized and NADPH is generated in this reaction, and that's why this pathway is known as this phase is known as the oxidative phase. And in the another phase, which is the non-oxidative phase, is associated with the synthesis of 5 ribose phosphate, which is used for the synthesis of nucleic acid as well. So we will talk about both this pathway in detail, the how it process and why it is called oxidative or non-oxidative. So first we will start with the oxidative phase. So as you know, the oxidative phase means so we have the glucose 6 phosphate, which is released by after the glucose enters the glycolysis. The first step of the glycolysis is the conversion of glucose to glucose 6 phosphate by hexokinases. So this first step is from the glycolysis, and once the glucose 6 phosphate is produced, instead of entering into the another step of the glycolysis, which is to produce fructose 6 phosphate, it enters the pentose phosphate pathway, and by the action of the enzyme glucose 6 phosphate dehydrogenase, it is a dehydrogenase which removes the hydrogen, and this enzyme uses NADP plus, and then we get 6 phosphoglucono-delta lactone. So it's a big name, but what is important to understand is that there are two hydrogens which are released, so from the first carbon here and here, So these hydrogens are released, and then we get NADPH and H plus. And then this NADPH can be used for the synthesis of the fatty acids. That's why this reaction or this phase is called the oxidative phase, because now NADPH is released. And now that we have the 6-phosphogluconolactone, then there another enzyme that act lactonase, which cause the the hydration, in which the one H2O is added, and what we get is 6-phosphogluconate with the carboxyl group. And then this 6 carbon is now reduced to the 5 carbon by the decarboxylation of the 6 carbon to form the D-ribulose 5 phosphate. And then this from the 6 carbon to 5 carbon is by the 6-phosphogluconate dehydrogenases. And during this reaction also we get NADPH, because this enzyme also uses NAD plus, and we get 1 CO2. And what we get is D-ribulose 5 phosphate, which is a 5 carbon instead of the 6 carbon. And then this can isomerize to form the ribose 5 phosphate pathway, and this ribose 5 phosphate can further be used for the synthesis of the nucleic acid, and it can also enter the non-oxidative phase where different sugars are produced, or where nucleic acid is produced. So here you can see, except the first step where the glucose 6-phosphate can enter the 6-phosphogluconate, or it can be reversible, but then the next steps are always irreversible. There is no reversible step, it's all irreversible reactions except the first one. So next step is the non-oxidative phase, where it's different. So in this phase mostly nucleic acids are generated and everything. So the ribose 5 phosphate, which we get from the last oxidative pathway, it enters the non-oxidative phase, where it can isomerize to different sugar, or it can combine with different sugar to produce different carbon sugars. So here you can see we have the D-ribose phosphate, which can combine or isomerize with 2-xylulose 5 phosphate. And when these two combine, this is all 5 carbon. So we get either 7 carbon sugar or the glyceraldehyde 3 phosphate. And then they combine to themselves and form this fructose 6 phosphate and a 4 carbon sugar. And then this 4 carbon combines again with 5, and it can also produce again fructose 6 phosphate and glyceraldehyde 3 phosphate. And then this glyceraldehyde 3 phosphate, fructose 6 phosphate, it can enter the glycolysis pathway. If you remember, there are different phases where one of the step is the production of the fructose 6 phosphate or glyceraldehyde 3 phosphate. So these two can also be feeded back into the glycolysis, and then glucose 6 phosphate produce this. So this is a feedback cycle where the glucose 6 phosphate produce ribose, and then it produces different products which can be feeded back into the glycolysis cycle, and then this cycle continues. So all this depends on the levels of the nucleic acids we have, the levels of NADPH. So I hope it's clear why it's called the non-oxidative, because in this reaction the nucleic acids are produced and also different sugars is produced. And this is oxidative because in this reaction mainly NADPH is generated. So how it is regulated? So the entry is controlled by the concentration of NADPH, as we have already talked. If you have higher concentration of NADPH, the glucose 6 phosphate will enter the glycolysis pathway, but if we have less concentration, then it will enter the pentose phosphate pathway. And the non-oxidative phase is controlled by the pentoses. If we have more pentose sugar, then then there is less non-oxidative. But if we need them, and if we need more pentose sugar, and then we get more non-oxidative phase. And the regulatory enzyme, the first two enzymes that we have talked about, the dehydrogenases, they are the regulatory enzyme, and then they depend on the NADP plus for their reaction, because then only this enzyme depend on them for their reaction. So they are also the regulatory enzyme in this pathway. And the NADPH, which is produced, it's a cofactor in the glutathione metabolism in RBCs, human red blood cells. So if we have less concentration of NADPH, then we have more injuries, or then it's difficult to keep up with the oxidative stress. With the oxidative stress, so NADPH is required for the RBCs. So I hope everything is clear. If you have any doubts, please let me know in the comment section, and please like, share and subscribe my channel. Thank you.